production in creation of proton gradient, flow of proton. Now actually first of all the proton gradient is required to be created. After that only the protons will flow. So creation of proton gradient and flow of proton. Clear? Now we have to see how in photosynthesis proton gradient can be created, and secondly what will happen when proton flow starts. So I have to rub it out now. So this thing according to chemiosmotic hypothesis was proposed by Peter Mitchell. Energy production in by synthesis in both photosynthesis and respiration involves two steps. First step, proton gradient is to be created, which is called creation of proton gradient, followed by flow of proton. Clear? Now I have to rub it out.
Sir, একটু sir.
Okay. So creation of proton gradient and then flow of proton. There are two basic points here for chemiosmotic hypothesis. First of all, proton gradient is to be created, and secondly, protons require to flow. Now I am going to rub it out. I am rubbing it out now. Look. This is the luminophthalate. Now you see this is the thylakoid membrane. So what happens if this is the PS2? And when photons hit P700 profile A, it gets energized to the release of one electron and it causes the splitting of water to form oxygen and hydrogen ion. Now the primary acceptor, you see what happens here. Primary acceptor the electron ejected from P700 profile A, P680. P680 profile A, this is the primary acceptor. Outside the primary acceptor, it lies outside the outer membrane of thylakoid. Primary acceptor.
See, figure.
ফিগারে না আমি একটু করে দিচ্ছি আগে প্রথমে দেখ PS2 is outside the outer membrane of thylakoid. From here electrons are carried to the hydrogen carrier. And hydrogen carrier, it is electron 2. Electron carrier that lies inner side of while carrying electron to the inner side of thylakoid membrane, it also carries protons along with it. That means a carrier that lies inner side proton comes out into the release into the inner side Look at this. What I am saying, the primary acceptor of PS2 lies outside the outer membrane of thylakoid. primary acceptor of pH2 lies outside the outer membrane of thylakoid, so that from there the electrons are required to be carried to the next carrier via stroma. So no electron carrier is present in the stroma. So a hydrogen carrier is required in this case to carry electrons to the next electron carrier. Is it clear to you? Or any doubt? দেখো বুঝতে পেরেছি কিনা প্রত্যেক দেখে নাও outer side of the membrane outer side lies outer side of the membrane easier. It lies toward the outer side of the membrane, so that it carries electron to the hydrogen carrier, not to any electron carrier. So hydrogen carrier, while carrying electron, it carries proton also, which is taken from the stroma and then drained into the lumen of thylakoid. So that this is the first how concentration of proton increases in the grana. Clear? Because here proton is pumped out into the stroma. Clear? There was doubt আছে কিনা Is it clear to you? How is proton gradient developed? This is the first point. As the primary acceptor lies towards the outer side of the thylakoid membrane, so from there the electrons are required to be carried to a hydrogen carrier for being carried to the next electron carrier that lies inner side of the membrane. The hydrogen carrier, while carrying electron, it carries proton also. These protons are taken out from the stroma and then it is ultimately released into the inner membrane or the what is called the lumen of the thylakoid. In this way, concentration of proton increases there. See the how it is coming. Clear?
Yes, sir.
Okay.
স্যার
Yeah.
স্যার যেখানে পিসি লিখেছো
হ্যাঁ
এটার আগে স্যার গুড একটা বাক্স আছে নিরবিধি কি লিখেছো?
cytochrome B6 and cytochrome F. Cytochrome B6, cytochrome F. Clear? Is it okay now?
আর স্যার NADP প্লাস যে লিখেছো এটার নিচে স্যার ENR নিচে
কোনটা রে আবার বলতো
NADP প্লাস সাইডেরটা একদম
FNR FNR means FD NADP reductase enzyme. FNR means FD NADP reductase enzyme. Clear?
আর স্যার PQ নিচে কি লিখেছো?
হ্যাঁ
PQ নিচে
PQ নিচে লেখা আছে PQ এরকম আর কি PQ after receiving proton and already electron from here, it becomes PQH2 by donating electron and proton. For the reduction of antiplast to form look at this. And I will do NADPH to H plus. So that way, because you know FT NADP reductase, you know, is responsible for the reduction of NADP, and these electrons and protons are required. The electrons are coming from the grana, but protons are coming from the stroma. As protons are taken out from stroma, means concentration of proton increase in the grana. And these three ways, proton gradient is developed across the thylakoid membrane from grana, but from thylakoid lumen to the stroma. দেখো বুঝতে পারছি না তিনটা ওয়ে Is it clear?
Yes, sir.
Okay, thank you. I will rubbing it out. So once gradient is developed, now what happens? From here, protons are carried into the lumen of stroma, and as a result of which proton gradient is broken down. Once proton starts to flow from the lumen of thylakoid to the stroma, along the gradient is broken down. Once the gradient is broken down, then what happens? And the enzyme present here in this is the ATP synthase. In the CF1. So what happens? That whenever proton starts to flow, or flow of proton starts from lumen of thylakoid to the stroma, then gradient is broken down so that the energy produced is responsible for the conformational change of the ATP synthase or required to activate ATP synthase enzyme to help in the conversion of ADP and PI to ATP. This is how ATP synthesis takes place according to chemiosmotic hypothesis as proposed by Peter Mitchell. There are more doubts here. Doubt here. list, please? Can anyone please? So this is what about the chemiosmotic hypothesis. I think it is clear to you all. Now coming to the dark phase of photosynthesis. So what is required here? Chemiosmosis requires one membrane is required, one proton pump is required, one ATP synthase enzyme is required, a proton gradient is to be developed. So all these things are required here. So this is what about the chemiosmotic hypothesis. Do one thing, please. You can just open your book and page 213. You see that is chemiosmotic hypothesis written here. Please go through this now. Page 213. NCERT book, chemiosmotic hypothesis, and just go through this line up to the 215, first two dangers. Please, I am giving you 2 minutes. Go through it now, everyone, please. Did you go through the book here?
Yes, sir. Yes, sir.
Okay.
Okay, sir.
Okay. Dark phase occurs in the stroma of chloroplast. In dark phase actually is called biosynthetic phase. Why it is called biosynthetic phase? Because in this phase food is prepared. And as I said earlier, it occurs in the stroma of chloroplast only during daytime because light phase and dark phase are interdependent. So the most important pathway of the dark phase is the Calvin cycle. Now we have to discuss the Calvin cycle first. There are some other cycle also, Hatch-Slack cycle, but first of all to Calvin cycle. And where it is found, it occurs in all photosynthetic plants. It occurs in all So Calvin cycle occurs in all photosynthetic plants. That means whether they are C3 plants or C4 plants. Actually, there are two types of plants. C3 plants, primary acceptor of CO2 is RUBP. In C4 plants, primary acceptor of carbon dioxide is oxaloacetic acid. Phosphoenol pyruvic acid. So primary acceptor varies. First stable product PGA, a 3-carbon compound. Four-carbon compound. So there are C3 plants and C4 plants, but you remember this, both C3 plants and C4 plants, and even in CAM plants, other plants you will get called CAM plants, in all Kelvin cycle occurs. That means Kelvin cycle is a must, because to prepare glucose, prepare food, without Kelvin cycle, food cannot be prepared. Without Kelvin cycle, food cannot be prepared. That's why Kelvin cycle occurs in all photosynthetic plants. Remember these things. Whether it is Hansen-Slack cycle or C4 plants, C3 plant, or sometimes CM plants, whatever it is. If food is to prepare in photosynthesis, obviously Kelvin cycle is required to occur. Is it clear to you all? Is it clear?
Yes, sir. Sir, what you written here? Primary acceptor of CO2 in.
Oh,
So in
primary
C3
acceptor
plants.
of CO2 is RUBP.
Okay, sir.
Here it is RUBP. RUBP. In this case, it is phosphoenol pyruvic acid. Phosphoenol pyruvic acid. Clear? Now, we have to proceed toward the Kelvin cycle. First step of Kelvin cycle is
Sir, in the brackets you have written 3C and 4C. What is the meaning?
3-carbon compound, 4-carbon compound. 3C means 3-carbon compound, 4C means 4-carbon compound. Clear?
Yes, sir.
First step is carboxylation. Carboxylation six RUB I'm rubbing it out, this part. So, RUB carboxylase is the enzyme. Both are taking place simultaneously. The same enzyme is same. This is called carboxylation. Clear? Or you can write in this way also directly, or suppose you can have 6 RUBP plus 6U2 plus 6H2O, RUBP carboxylase 12 PGA is also true, same thing. Clear? After that, so this is what carboxylation, they are the first stable product. the first stable product. After that, next part is reduction. What happens in reduction?
স্যার
Yes.
স্যার তুমি 6CO2 র উপরে RUBP যে লিখেছো এটার মানেটা কি?
The enzyme that catalyzes this reaction, RUBP carboxylase is the enzyme that catalyzes this reaction. Clear?
Yes, sir.
Okay. In reduction involves following steps. Reaction following one. (12 PGA) react with (12 ATP). The enzyme involved is kinase. Next, (12 DPGA). Just a minute. (12 NADP H plus H plus). (12 PGLD plus). (12 NADP plus). (12 H3PO4). Enzyme involved here is now next reaction.
Sir.
Out of 12 PGLD,
तो सेकंड रिएक्शन है 12 PGA তারপর কি লিখেছো স্যার সেকন্ড রিঅ্যাকশনের মধ্যে 12 PGA অ্যালডিন এটা
12 PGL phosphoglyceraldehyde plus 12 NADP plus 12 H3PO4. Now here out of 12 PGLD formed, these two PGLD give rise to glucose. How? I will show you. First reaction number 2, reaction number 3. 1 PGLD convert to dihydroxyacetylene phosphate. Enzyme is aldolase. Then next reaction 4 PGLD, other PGLD plus DHAP. Isomerase. Fructose 1,6-bisphosphate. After that, reaction number 5 I am writing here. Phosphate is the enzyme. This one phosphate group is liberated out. Reaction number 6. Seven. This is how glucose is formed from 2 PGLD. Is it clear to you? Look at this. Out of 2 PGLD, 1 PGLD gets converted to dihydroxyacetone phosphate. DHA means dihydroxyacetone phosphate. Now other PGLD left, this PLD react to this DHAP to form fructose 1,6-bisphosphate. And the enzyme is aldolase. From fructose 1,6-bisphosphate, one phosphate group is liberated out to form fructose 6-phosphate. Next reaction. Fructose 6-phosphate in the presence of phosphohexose isomerase enzyme, it is converted to its isomer glucose 6-phosphate. Lastly, from the glucose 6-phosphate, phosphate group is liberated out as H3PO4 and glucose is formed. Eventually, glucose gives rise to sucrose, starch, etc. So this is the second step, reduction. Clear? देखना वाला बोले. In reduction, a series of reactions are taking place to form glucose. Clear?
Yes, sir. Clear.
Okay. Now, the third step. I will rub it out, please.
Sir,
Can I rub
wait
it out
for a
now?
while.
Huh?
Sir, wait for a while.
Okay.
Yes, sir.
Now step 3. Step 1, carboxylation. Step 2, reduction. And step 3. Regeneration of RBP. What happens here? Different intermediate carbon compounds six RUMP. Next step, clear? In this way, again RUP is regenerated. RUP means ribulose monophosphate. RUP means ribulose monophosphate. So these are the 3 steps. Clear?
Yes, sir.
Regeneration of RUMP 2 steps. In the step 1, different intermediate carbon compounds will give rise to 6 RUMP, and then 6 RUMP gets phosphorylated in the presence of ATP to form 6 RUP. So that the reaction can continue. Calvin cycle can continue. Clear?
Yes, sir, clear.
Okay. Can I rub it out now?
Yes, sir.
Okay. I'm taking 1 minute more.
Sir, একটু
ঠিক আছে। Clear? Okay?
Done, sir. Yes, sir, done. Clear? This is the Kelvin cycle shown. Go through it, please. I am giving you 2 minutes, coming within 2 minutes. Complete it by this step. Now, question comes here. How many ATP molecules are required to produce 1 molecule of glucose? Honishika, how many ATP molecules are required to produce 1 molecule of glucose in Kelvin cycle?
Sir, can you repeat the question, please?
How many ATP molecules are required to produce 1 molecule of glucose in Kelvin cycle? Tell me.
Sir,
Hmm.
sir, I'm not sure about it, but for me, 18 ATPs are required. Maybe. I'm not sure.
Can you explain how? 18? Can you explain how it is 18?
Sir,
You are
I
absolutely
told
right.
only I'm not
12 ATPs are
sure.
here, 6 ATPs are here. In these are 18. 12 ATPs are required to convert PGA to DPGA and 6 ATPs are required to convert RUMP to RUBP. Clear? 12 ATPs are required to convert 12 PGA to 12 DPGA and 6 ATPs are required to convert RUBP to RUMP. Clear?
Yes, sir.
Okay. Next, similarly, question comes. How many NADPH plus H plus are required to produce 1 molecule of glucose in Kelvin cycle? How many NADPH plus H plus molecules are required to produce 1 molecule of glucose in Kelvin cycle? Then what's the answer?
Sir, 12.
12. Yes. Other questions from here come. How many turns of Kelvin cycle are required to produce 1 molecule of glucose? How many turns of Kelvin cycle are required to produce 1 molecule of glucose? How many turns?
Two.
How many turns of Kelvin cycle are required to produce 1 molecule of glucose? The turns is, you see, in one turn, in one turn of Kelvin cycle, one molecule of carbon dioxide gas fixed. Remember this thing, in one turn of Kelvin cycle, there is fixation of 1 molecule of carbon dioxide. Clear? If it is so, how many turns required for the fixation of 6 molecules of carbon dioxide? 6 turns. So remember this thing, write down, in one turn of Kelvin cycle, there is fixation of 1 molecule of carbon dioxide. Write down, in one turn of Kelvin cycle, there is fixation of 1 molecule of carbon dioxide. So it's
Sir, can you repeat it again? Sir,
write down.
1 turn of Kelvin
In
cycle.
1 turn of Kelvin cycle, there is fixation of 1 molecule of carbon dioxide. Okay?
Yes, sir.
6 molecules of carbon dioxide are required to be fixed to produce 6 molecules of carbon dioxide are required to be fixed to produce 1 molecule of glucose in Kelvin cycle.
Sir, can you repeat again?
Write down. 6 molecules of carbon dioxide are required to be fixed to produce 1 molecule of glucose in Kelvin cycle.
Sir, site.
Hence, 6 turns of Kelvin cycle are required activator. Hello? Clear? To produce 1 molecule of glucose in Kelvin cycle. 80 molecules required to produce 1 meter. Is this clear? So 6 molecules of carbon required to fix to produce 1 molecule of glucose in Kelvin cycle. Hence, 6 turns of Kelvin cycle are required to make 1 molecule of glucose. Hence, 6 turns of Kelvin cycle are required to make 1 molecule of glucose. Clear?
Yes, sir.
Okay. So this is what about, so you need to remember the whole cycle actually. Questions may come from any part of it. So this NADP, because what I'm telling you, this NADP required in the light phase. In this way, light phase is also found to be dependent on the dark phase. Are you getting me? The ADP and NADP produced in the dark phase are required to carry out the light phase. And in this way, actually these two phases are found to be interdependent because the ATP and NADPH phase plus produced in the light phase are required in the dark phase. Similarly, ADP and NADP plus produced in the dark phase are required to carry out the light phase. In this way, these two phases are found interdependent. Clear?
Yes, sir.
Okay. Can I wrap it out now? Is it clear to everybody what is Kelvin cycle? Generally, question comes, to fix 1 molecule of carbon dioxide in Kelvin cycle, listen carefully. To fix 1 molecule of carbon dioxide in Kelvin cycle, how many ATPs and NADPH phase are required? To fix 1 molecule of carbon dioxide in Kelvin cycle, how many ATP and NADPH phase molecules are required? Can you tell me? In order to fix 1 molecule of carbon dioxide in Kelvin cycle, how many ATP and NADPH phase molecules are required? Anyone, please?
Sir, 24.
24, how can it be? I am repeating the question again. How many ATP and NADPH plus molecules are required to facilitate fixation of 1 molecule of carbon dioxide in Kelvin cycle? To facilitate fixation of
1 molecule of carbon dioxide in Kelvin cycle. Clear? Okay, any doubt? Is it clear? What is the answer?
So 3 ATP.
Answer is 3 ATP and in Kelvin cycle. 3 ATP molecules and 2 NADPH molecules are required to fix 1 molecule of carbon dioxide in Kelvin cycle. See, in these way questions are coming in the entrance. So how many ATP molecules are required and how many NADPH molecules are required to fix 3 molecules of carbon dioxide? It is written 1 molecule. Sometimes it comes 3 molecules of carbon dioxide. Then obviously there should be in 1 molecule 3 ATP means 3 molecules 9 ATP. Okay. So in this way the questions are coming actually. So you to confuse you actually. So remember this thing. To fix 1 molecule of carbon dioxide, there is requirement of 3 ATP and 2 NADPH. In this way, it is 3 molecules, 4 molecules, 5 molecules, 6 molecules. You can easily determine these things. And in this way, questions are actually coming. There, how many ATP molecules are required? In this case, how many ATPs are required to fix 1 molecule of carbon dioxide? Sometimes comes 2 molecules of carbon dioxide, 3 molecules of carbon dioxide, even given the 4 molecules of carbon dioxide. That means up to 6. Everyone knows how many ATP molecules are required to fix 6 molecules of carbon dioxide, 18. But sometimes whenever the questions given like this 4, 5, sometimes actually some mistakes are carried out that you are writing suppose a wrong, you are giving wrong answer actually. That's why you have to remember this point first. So yes, 3 ATP molecules are required and 3 ATP and 2 NADPH are required to fix 1 molecule of carbon dioxide. Then easily you can answer what about the number of carbon dioxide fixed in this case. So this is most important thing. Remember this thing, in Kelvin cycle everything is okay. Kelvin cycle is required. And here also RBP actually is to receive carbon dioxide. Divalent bisphosphate occurs in the stroma only. Now remember this thing. The basic problem with this is that if intensity of light is more, then there will be the rate of light phase increases, then there will be more and more, there will be production of more and more oxygen. So relative concentration of oxygen increases. And then what happens? RBP is found to react to the oxygen and photorespiration is initiated. They are just giving you one simple hint today. That sometimes what happens in this case, if intensity of light increases, then the rate of photosynthesis increases, light phase increases, so there will be more production of oxygen. If there is more production of oxygen, the consequence is that RBP is found to react to the oxygen instead of reacting with carbon dioxide. If RBP is to react with oxygen, this is the beginning of photorespiration. And obviously in photorespiration, photosynthetic activity decreases. So this is actually a photosynthetic loss occurs. Because why it is called respiration, I will be showing you next day that here CO2 is produced as a byproduct. Is it clear? Are you getting me now?
Yes, sir.
Okay. So that's all for today. In my next class, I am to start with I am to complete this chapter actually. I am to discuss photorespiration, I have to discuss C4 pathway, I have to discuss CM, and I have to actually discuss the factors affecting photosynthesis. Okay?
Yes, sir.
Okay, thank you very much.
Sir,
Bolo.
Sir, you have told that from January you will start offline.
So from January means January 15, I've told you.
Yes, sir.
If everything okay, then there is intention to start after January 15. Then 16 or 17.